I'm Daniel Levine, and this is the Bio Report. Type 1 diabetes is still a disease we manage day to day rather than prevent, and today's approaches do little to stop the immune system from attacking beta cells in the first place. Zag Bio is trying to change that by turning the thymus, the body's training ground for T cells, into a therapeutic engine for immune tolerance, using thymus-homing antibodies to program long-lived antigen-specific Tregs against type 1 diabetes and other autoimmune diseases. Zag Bio CEO Jason Cole discusses type 1 diabetes, how the company's thymic training platform fits into the emerging tolerance landscape alongside cell therapies and nanoparticle approaches, and the potential to extend this strategy to a broader set of autoimmune diseases. Jason, thanks for joining us.
Danny, happy to be here. Thanks.
We're going to talk about type 1 diabetes, Zag Bio, and your efforts to develop therapies that target the thymus gland for type 1 diabetes and other autoimmune conditions. Perhaps we can start with the thymus gland. What role does it play in the immune system?
Great question. And, the thymus is a fascinating organ that, to be honest, I wasn't very familiar with until I joined Zag Bio about 9 months ago. But what the thymus does is it essentially trains your T cells to be your immune system. And it trains those cells by having, naive T cells enter the thymus where they're exposed to antigens for every tissue in your body. And so those naive T cells, if they have high affinity for antigens that are that are your own tissues, they're actually destroyed because those would become autoreactive T cells, which would be bad to have circulating. And a lot of this happens, early in childhood when you're an infant. But importantly, the thymus also creates T regulatory cells, or Tregs. And Tregs are essentially the diplomats of your immune system. They traffic to places where there's inflammation, and they can calm down an immune attack that is ongoing. And so what's really cool about the thymus is we can leverage it therapeutically now by introducing antigens that may be under attack by your own immune system. So autoimmune disease, right, is, as the name implies, your own body going after your own tissues. So we figured out how to cleverly leverage this unique organ to create these long-lived T regulatory cells in people, in vivo.
So is the thymus implicated in autoimmune diseases like type 1 diabetes, or is it just a matter of leveraging its way to turn off T cell activity against a specific cell type?
Yeah, so it's more the latter. So the thymus itself doesn't, cause autoimmune diseases, but it has this sort of capability to make these long-lived Tregs against other antigens, which is what we're really trying to leverage it for, which is something that is relatively new. The thymus is having a bit of a moment based on some more recent, emerging research.
We mentioned your initial focus is on type 1 diabetes. Can you explain the difference of type 1 from type 2 diabetes and how it's an autoimmune disease, whereas type 2 isn't?
That's right. Yeah. So the end state is, bad in both, right? Where you are, dependent on insulin to moderate your blood glucose, but the biology is fundamentally different. So as you mentioned, type 1 diabetes is an autoimmune disease where your own T effectors or T cells attack the beta cells in your pancreas. And so those beta cells in healthy individuals make insulin, which moderates your blood glucose. And failure to make insulin can lead to really high blood sugars, which in an acute moment can cause something called diabetic ketoacidosis, which could kill you. But most in America, there are ways to get sort of diabetes under control. If you end up in this state, but the long term impacts of type 1 diabetes are heart failure, kidney failure, vision problems, all sorts of other diseases. And so for type 1, this autoimmune attack just wipes out those beta cells and you end up insulin dependent. Type 2 is really driven by a number of factors. So exercise, diet, genetic factors, which cause the pancreas and those beta cells to just get worn out and to sort of lose their capability to control your blood glucose, which puts you in the same place.
How is type 1 diabetes generally treated today, and what's the unmet need that still exists for people living with the disease?
Sure. So there's still really significant unmet needs. So there are like 1.6 million Americans with type 1 diabetes. There are 9 million, throughout the world. And like any disease, there are different stages. So if you think about cancer or other diseases. So stage 4 type 1 diabetes is when your beta cells are gone, right? So you are insulin dependent. And, in the old days, you would do sort of a manual injection by a syringe at mealtime. What's happened now is there are insulin pumps that are connected to continuous glucose monitors, either through a phone or some other device that essentially releases insulin on a continuous basis to control your blood sugar. But then the patient has to instruct around a bolus when they have a meal. And the whole goal here is to keep your blood sugar within a tight window. Because if you fail to keep your blood sugar within that tight window, you can have these long-term complications from type 1 diabetes that I mentioned earlier. So although pumps and continuous glucose monitors have been a significant improvement, compliance with everything you need to do on a day-to-day, hour-by-hour basis is not perfect, especially when you think about kids, right? So type 1 diabetes was historically called juvenile diabetes. And so a 4-year-old and them managing their pumps and sensors is a lot harder, even with the most conscientious parents. And so for stage 4, that is, what you want to try to replace. And there are companies that are working on cell therapies that would make insulin in your body to replace those. That's not where Zag is going, but that's part of the treatment. Stage 3 type 1 diabetes is newly diagnosed, right? So your blood sugar is going up, you're symptomatic, but you still have some beta cells left. This is often called the honeymoon period, where you're still making some insulin. And so there are a number of companies developing therapies to extend that period of time or delay getting all the way to full-blown diabetes, stage 4. That's an area where I think we will be conducting clinical trials to prove our mechanism and how our therapy works. But Zag is particularly interested in getting into stage 2 or stage 1 type 1 diabetes. And that's where you have these elevated autoantibodies that can be detected with a simple blood test that shows the autoimmune attack has started, right? The beta cells are being, attacked. And we could then intervene with our therapy, get these special Tregs to home to the pancreas and then protect those beta cells and prevent type 1 diabetes from happening at all. And so, our goal is to make sure there are not future type 1 diabetes patients by being able to intervene early and resetting the immune system so it stops attacking your own beta cells.
You've helped build multiple biotech companies, but Zag seems like it may be more of a personal effort because you live with type 1 diabetes yourself. What has it been like to live with the condition, and what role did that play in your decision to become CEO of Zag Bio?
Yeah, great question. A short answer is, this time it's personal, but the longer answer is, yeah, I was diagnosed late in life. I was diagnosed in my late 40s. I have a family history of type 1 diabetes. My dad was diagnosed in his 30s. He lived with it for 40 years, but ultimately passed away from a lot of those, comorbidities, as they say, that come with type 1 diabetes. So, like any person living with a chronic disease, I have my good days and my bad days managing it. But as it relates to Zag, I've been fortunate to work in this industry for over 20 years. I've learned a lot about how to operate a biotech company, how to advance therapies from research all the way to the market. And so for me, right, this is an opportunity to really bring those skills to bear on a disease that is really important to me and my family, but also, millions of patients around the world. Throughout my career, I've always sought to connect with the patient communities that we're trying to help. And so in that case, Zag is no different, but it was the kind of opportunity that'd be really hard to turn down to help future generations of type 1 diabetics.
As you've said, this is also known as juvenile diabetes. Is it unusual to be diagnosed at age 40 with this condition?
It is pretty unusual. So, as I've learned both as a patient and an executive, the reason we don't call it juvenile diabetes anymore is it is diagnosed kind of throughout life. I think just under 50% of patients are actually diagnosed in adulthood, and diagnosis rates are going up. With respect to type 1 diabetes. So, my particular circumstance is particular to me, but it is still, a disease that's sadly growing that we need to come up with some therapies to prevent.
And being someone with a condition, how has that influenced the way you think about things like innovation in autoimmune disease and clinical trials?
Sure. So, in general, I think we're living in a time of just incredible innovation in the biotech space. And we've seen a lot of progress, even in the past like 5 to 10 years in a lot of areas where the standard of care just shifts overnight. There are a number of cancers that used to be, this terrible sort of death sentence that now are strangely manageable, right? Where with certain red targeted regimens, you're able to have lasting remissions. There are a number of rare diseases now where therapies exist that are essentially curative. And you look at the GLP-1s and what they've done for type 2 diabetes and obesity. So, I'm a believer that autoimmune disease is one of those areas that's ripe for that same kind of innovation. As we discussed, T1D doesn't have a lot of innovation on the therapeutic side, right? Pumps and sensors are medical devices, right? And Zag, like a number of other companies, are joining the sort of group of companies that are really trying to bring that transformative innovation to immunology by addressing the root causes and really resetting your immune system to make these diseases go away. So as a patient, I'm excited about it. As a biotech CEO, I'm doubly excited about it because of what it could mean for our company.
There is growing interest in immune tolerance from cell therapies to antigen specific approaches. Where does Zag fit within that landscape?
Yeah, great question. So we should probably talk a little bit about what tolerance really is and what it means. So, in simplest terms, immune tolerance is just having this balance with your immune system where it can still function to get rid of foreign things like infections or other intruders, but then it also isn't going haywire to make you sick with sort of an autoimmune attack. So the idea around tolerance is with different therapeutic approaches trying to restore that balance. And as you mentioned, there are folks who are working in this space that are doing what we call peripheral tolerance. So what does that mean? They are using nanoparticles or other approaches to wipe out T effectors that are, having this autoimmune attack. And that's called through like deletion or anergy is how they describe that. And then what that means is regular Tregs can then sort of do their, suppressive job. They're cell therapy companies, right, who are engineering these Tregs outside the body to behave in an antigen specific way inside the body to protect the tissues that might be under attack. What Zag is doing, I think, is a little more elegant. So we have these antibodies that home to the thymus, to a specific target, and an antigen presenting cell, where we present the antigen for the naive Treg to get trained to protect and then go out into the system, find the tissue that's under attack, and then protect it there. And so we believe our approach is differentiated because those thymic Tregs, the native Tregs, are really long lasting. So in humans, they can last decades, and they also stay Tregs. Sometimes when you are manipulating other T cells to become Tregs, maybe in a transitory phase, they can revert back to the T effector phenotype, which is back to attacking. So again, this gets to duration of therapy, which is something we're really excited about.
How do you differentiate Zag's approach from broader immunomodulation strategies being pursued today?
Sure. So, in general, right, historically, we've treated autoimmune diseases with broad immunosuppression, right? So it's the sledgehammer approach to sort of bring everything down. And you can listen to a Super Bowl commercial and hear all the side effects of that. And sadly, broad immunosuppression also hasn't necessarily been able to help all patients, right? There are many patients who don't respond. There are many patients who relapse, from these therapies. And so, Zag is part of a number of companies that are really trying to do more targeted, long-lasting approaches to autoimmune disease. As I mentioned earlier, there are people working on tolerance through using different modalities. But I think our differentiation is creating this really powerful cell type using an in vivo approach with our antibody. I'm sure you and others follow, the CAR T space, right? So these were engineered T cells that have been used to attack cancer, primarily in the blood. And a number of those first CAR T programs were ex vivo, right? There were the patient's own cells that were manufactured to then turn those T cells into killers of tumors. Now there are a bunch of companies that are doing in vivo CAR T, right? They're delivering the sort of toolkit to turn those T cells into attackers in the body. We're doing something analogous by using an antibody to train Tregs to, in this case, be protective, to be those diplomats of the immune system.
And do you think immune tolerance will become a new pillar of autoimmune treatment alongside biologics and cell therapies?
Uh, I believe so and hope so, mainly for patients and also for companies like Zag. Now, it's not going to be, one therapy that covers every autoimmune disease. So, autoimmune diseases are very idiosyncratic and specialized in terms of, how they impact patients. But for companies that can really unlock tolerance, there is a potential to, knock these diseases off one at a time and really improve standard of care in a lot of places. So I'm a believer.
Well, let's talk about your lead experimental therapy, ZAG 101. What is it and how does it work?
Sure. So, as I alluded to before, right, our technology platform is an antibody based platform. So, Zag 101 is a bifunctional antibody. So, not bispecific, but bifunctional. So, that means one end of the antibody targets these specific cells in the thymus. So, it gets to the thymus and then it gets to those cells where then it can present an antigen. The other side of the antibody is the antigen. So in the case of type 1 diabetes, it's an antigen that is expressed in the insulin producing beta cells. And so that antigen is what those Tregs are then seeking to tolerize against, to protect. And so then those newly trained Tregs, if we get the dose right, will then home to the pancreas where there's this, inflammation happening from your autoimmune attack, and it's going to calm things down, right? And so, our therapy, it is an antibody, but it's not directly therapeutic as the antibody. It's an antibody to induce a Treg population, basically using the thymus as our bioreactor. And so that mechanism is really, cool and interesting, but that's what our therapy is.
And is it expected that this would require repeated dosing, or is this a one and done therapy?
Yeah, it's a good question. So we are aiming for these to be really long-lived, durable therapies. So words like one and done and curative, I think are a little loaded terms, especially when you think about how complex, immune diseases are. But I think as I mentioned earlier, Tregs last decades, that population in humans. If you think about it, our immune systems or the immune systems of healthy people were trained when you were a kid and they're still doing their job. So if our antibodies can induce this antigen specific Treg population, it should last for a very long time. Our therapies would be redosable. That is a feature, not a bug. And so, what would happen in a disease like type 1 diabetes is you could take that blood test I mentioned, that screening blood test, maybe a year after getting our therapy, and we would see if those antibodies are rising again. Or you could take a blood glucose test and see if that's rising again. So, if that's happening, maybe the autoimmune sort of tide is turning against you. You could redose and then tamp everything back down again. So we're going to learn in the clinic the real kind of pharmacokinetics and pharmacodynamics of our therapy. But I certainly believe and aspire to, based on data, that this is, a long-lasting therapeutic intervention that you might need to top up the tank over time.
Well, what's known about 101 from the studies you've done preclinically with regards to its safety and efficacy?
Sure. So, we're entering the clinic later this year with Zag 101. As you would expect, we've done, a battery of preclinical and safety assessments of the molecule. And so, on the efficacy side of things, we've seen really nice efficacy in multiple animal models. And then we're completing our tox studies just to make sure we feel like it's safe to enter the clinic, again, starting at low doses. I think some of the most exciting data we've seen preclinically is in these mouse model of diabetes called the NOD model. So this is non-obese diabetic mice. So these are these mice that spontaneously get something that looks like type 1 diabetes. And so we've used this model to do some interesting studies to prove whether our therapy has bystander tolerance. So Tregs, another key feature of Tregs is they certainly play this sort of diplomacy, immunosuppressive effect by themselves. But once they're in tissues and once they're encountering inflammation, they release all these cytokines to recruit other T cells to the fight. And that's what they call bystander suppression. And so what we figured out how to do is by targeting a single antigen, we are able to recruit other, T cells to this fight that can also suppress inflammation in cells that express a different antigen, right? So in a disease like type 1 diabetes, there are multiple antigens that are implicated in the autoimmune disease. And if you want to have a long-lasting effect, you have to cover them all. And there are companies that are putting all four antigens into a single nanoparticle to try to get after all of them. What we're able to do with this concept of bystander suppression is by targeting a single antigen, but then leveraging this bystander effect still have an immunosuppressive effect across all those different antigen-specific cells. So that's some of the most exciting data we've had preclinically, which gets physicians and key opinion leaders excited about moving this into the clinic.
And how are you thinking about inclusion exclusion criteria and your primary study endpoints?
Yeah, so we, I think, are fortunate to be able to start our study in patients with type 1 diabetes. We'll be starting in adults because you start in adults for safety reasons. And we'll be starting in adults with stage 3 disease, right? So they're folks who are newly diagnosed, they're starting to take insulin, but they still have beta cells to preserve. And so, one of the key endpoints in type 1 diabetes drug development is C-peptide, right? So, this is a measurable biomarker that indicates your beta cell's ability to keep making insulin. So, every patient we enroll will have a minimum level of C-peptide that we are going to seek to preserve, right? So, if our drug is working, the C-peptide will not drop off a cliff because if you do not have beta cells anymore, you don't make insulin and you don't make C-peptide. So, that's going to be an important biomarker for us to track in this first in human study to get a sense of efficacy. We're also going to be leveraging some other biomarkers where we're going to measure these T cell populations in blood. So that means these T effectors, right? We want the antigen specific T effectors that are doing the damage to go down. And we want to be able to detect our antigen specific T regulatory cells. And so these are also some biomarkers we're going to track in these patients. But importantly, this study is about safety and dose in the first instance. And so we're going to start our study with single doses that are going to go up safely and gradually to eventually figure out the right course of treatment and exposure we need to have the kind of efficacy we want. And so once we complete our sort of single dose escalation, that'll allow us to pivot into perhaps a multi-dose approach or a single dose approach that we would then move into further patient populations. Importantly, if we have nice safety in adults, we could start moving into pediatrics or adolescent patients.
How are you thinking about demonstrating disease modification rather than just symptom control?
So this comes down to that C-peptide endpoint I mentioned before, right? So, symptom control in the case of type 1 diabetes is about like HbA1c and glucose, right? That is the ultimate symptom of not having insulin producing cells. But, with our aspiration to really have profound disease modification, it is simply following the C-peptide signal for longer and longer periods of time. And if our hypothesis around antigen-specific Tregs is true, then that C-peptide should just stabilize in adult stage 3 patients, in adolescent stage 3 patients, and then eventually, if we move into stage 2 patients, right, who have yet to flip to the more severe forms of type 1 diabetes, we're just maintaining that immune sort of homeostasis so they never have a dip in C-peptide. There's a lot of, clinical trial ahead of us to prove that, but that's the idea.
You talked about autoimmune diseases being idiosyncratic, but beyond type 1 diabetes, what other conditions are most compelling for this thymus targeting approach?
Yeah, so that's another reason I was really excited to join Zag is now that we've figured out how to target the thymus with an antibody that is systemically administered, so an IV or a subcutaneous injection. For other diseases, we then just have to focus on swapping out the antigen, right? So, what we are getting the Tregs to tolerize against. And so, two areas of interest to us that our research team is focused on right now are diseases of the gut so ulcerative colitis and Crohn's disease, as well as rheumatoid arthritis and other, synovial diseases. And these are areas where there is a lot of research and development ongoing. There are a lot of clinical programs that are really trying to help patients, but they're also largely still either immunosuppressive or they don't have broad population effects, or they will be needed to be administered in combination. If you believe, like we do, that Tregs have this long-term potential to reverse or restore, reverse autoimmunity or restore, immune function, these would be tremendous diseases to go after next.
You previously held executive positions at Bluebird Bio and Salugen. What did you learn there that you're applying directly to how you're building Zag Bio?
Yeah, Danny, it's funny. You're the product of your career and the things you learn, over the course of your career. And you're also the product of, the time in which you're working. And so in the context of 2026, where it is challenging to fund, preclinical biotechnology companies, the lessons I'm applying now are about advancing therapies that are truly impactful and differentiated, and doing so in a very focused and capital efficient manner. So, with all the other companies I've worked at, we've tried to bring things that are, you know, curative or have high potential. And the real challenge in this environment is to do so with limited resources. So at Zag, we have a very small and focused team that is working thoughtfully with a lot of external vendors to help us execute our preclinical development and then our clinical development going forward. And I think that's the sort of model for biotech in the US that a lot of companies are following. And then if you see that translation, if you see that signal in the clinic, you've basically earned the right to bring in more capital and run that next clinical trial or to expand your pipeline further. So it's harder, but I think there's real discipline with trying to do this with limited resources.
Zag recently raised an $80 million round that included participation from several high profile strategic investors. What seemed to resonate most with investors like AbbVie, Regeneron, and Sanofi?
Yeah. So, I was just talking about limited resources. And then, we have raised $80 million. And I think we're fortunate to have a great syndicate of investors who, had the, vision to support a company that's going after this, audacious goal of, long-term, immune tolerance. But yeah, with respect to AbbVie, Sanofi, and Regeneron, these pharmaceutical companies have the luxury in a way to scan the horizon, right? To look forward a decade and try to figure out what standard of care going to be then. And a lot of these companies have their own immunology franchises, right? That make billions of dollars that will eventually go generic or could be disrupted by a shift in the standard of care. And so I think, each of these companies has their own strategy in I&I, as we call it. But I think the reason they're interested in Zag is we're at the forefront of immune tolerance, and we're at the forefront of applied therapeutics for Tregs, right? We're not engineering them outside the body, we're making them in vivo. And so by making an investment in Zag early on, they get to hang around the hoop and have a seat at the table and watch as we progress not only the T1D program, but our research in other diseases that might be of interest to them. And so I'm fortunate to have those investors in the syndicate and hope to get their input as we continue to advance as a company.
And how do investors view your own relationship with type 1 diabetes? Do they see that as a plus, or does that concern them about your ability to make dispassionate pipeline decisions?
Yeah. So, it's not something we talked about a lot in like the interview process or since I've joined. But what I would say is, investors in early stage biotech companies, when they're hiring a CEO, they're looking for someone with passion and drive to move, the science and the programs forward with real commitment and real focus. And so, I think my own personal and family link to T1D certainly provides some fuel for that drive. But to be honest, I've always been really patient centric at all the other companies I've worked at over time across a lot of diseases, which, I don't have a direct sort of connection to. And I think part of that, comes from, I think, the culture that a lot of companies here in the biotech field have. So, Roy Vagelos, the CEO of Merck for many years, and Henry Termeer, the CEO of Genzyme, here in Cambridge, have both been credited with saying that biopharma companies should always put the patient at the center of their decisions, and that any when you do that, any success that follows, should be your North Star for how you make those decisions. And I agree with that. I subscribe to that approach. And so I have my own sort of patient perspective, but I'm just one patient. And so at Zag and other companies I've worked for, I've always tried to sort of bring those perspectives into the mix so we can make the right decisions for the patients, which will ultimately be the right decisions for the company. I'm also a very data-driven person. And so when you're making those hard decisions about advancing a program, stopping a program, in my experience, right, truly understanding and reading the data objectively also gets you to the right place. So I'm excited to, continue this journey with Zag and try to have that kind of impact, not only in T1D, but in a number of diseases.
Jason Cole, CEO of Zag Bio. Jason, thanks so much for your time today.
Sure. Thank you. I really enjoyed it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.